Hello all, welcome back again to the YouTube channel One Mark Solution. Now I'm here with few questions, assertion reason kind of question from the chapter 9, strategies for enhancement in food production, 12. First question, assertion states like this. Hybrid seeds must be produced every year to obtain maximum advantage of heterosis. So what is heterosis here? Maximum advantage of heterosis. So when we say this, improved or increased function of any biological quality in the hybrid than the parental generation is called as heterosis. Okay, fine. So the statement is very correct. Now let us see the next statement. Heterosis is lost by inbreeding. Again, it is true. Inbreeding improves pure lines and the heterosis vigor is lost by this. So let us see that this due to inbreeding only every year or after each generation new seeds are produced. So both the statement are correct and this term inbreeding gives a proper explanation for this. So the option is A. Okay, let's go to next question. Virus-free plants can be produced from virus-infected plants by means of meristematic culture. No doubt that is correct. So we know that using meristem culture, usually we produce that. And so what is this? The capacity to create new organism from a single cell, which plants possess. That is the totipotency. And this culture is based on the totipotency. So viruses, the second statement say that the virus fails to grow during the growth of the host in an artificial medium. Is this correct? It is not like that, isn't it? Actually, viruses spread systematically in the whole plant except the meristem, because the molecular and cellular mechanism in the meristematic cells stays unaffected. It gives a protection and because of that, it stays unaffected. So which is the option here? Here, assertion is true, but the reason is false. So the option C is correct. Let's go to the next question, that is question number 3. A gene bank should not be regarded as a plant museum. To understand this, first of all, what is a gene bank? So it is a repository of many biological material. It can be a repository of plant materials like collected, stored, that is seeds or germplasm, etc. And here the option, the reason statement is germplasm stored in the gene bank are actively utilized by breeders to develop novel varieties, of course. So whether during the plant breeding or animal breeding, the germplasm used and stored in the gene bank is used for that. That is a function. So both assertion and reason statements are correct. And the second statement gives a proper answer for the or explanation for the assertion. So the option is A. Question number 4. Protoplast fusion is one of the major advantage of tissue culture. So what is a protoplast fusion? Here actually two different plant varieties with the desirable traits can be, it is like crossed undergoing protoplast fusion. And the naked protoplast of two different plants fuse to form a hybrid. Yes, that is what happening. So the statement A, assertion is correct. The reason is also correct. But this reason, that is the naked protoplast of two different plants fused to form a hybrid, is not giving a proper explanation for the first statement. So the option is B. Okay. We have come to the last question from the same chapter. Somolonal variation occur in tissue culture process. So what is somolonal variation? So this is something. Somolonal variation is a variation seen in plants that have been produced as a result of tissue culture. And let us see the second statement. Variations cannot occur in nature. Even though the first statement is correct, the second statement, that is variation can definitely occur in the nature. So that is wrong. So the option is C. So that is the last question from the chapter, and we have come to an end of this. I'll come back again with the assertion and reason kind of question of the next chapter, that is chapter 10. Bye.